Hello, everyone. In this lecture, we will discuss how microbes establish themselves within a host after exposure. Every day, we encounter countless microorganisms, yet the vast majority never cause infection. Many land on tissues where they cannot survive or reproduce, while others are removed by our barrier defenses, including mucus, tears, saliva, urine flow, and normal immune surveillance. For a pathogen to cause disease, it must overcome these defenses and successfully establish itself within the host. This requires attachment to host tissues, persistence in the face of physical removal mechanisms, and in some cases, invasion of deeper tissues. Today, we will examine the strategies microbes use to overcome these barriers and begin the process of infection. By the end of this module, you should be able to explain why adhesion is required for infection and compare fimbrial and afimbrial attachment. So let's briefly review the stages of infection from the pathogen's perspective. First, the pathogen encounters the host. Next comes adhesion, where the microbe attaches to host tissues. After that, after that attachment, some pathogens invade deeper tissues, survive host defenses, cause damage, and eventually transmit to a new host. Notice that adhesion occurs very early in this process. If a pathogen cannot attach, the remaining stages often never occur. Our bodies are remarkably effective at removing microbes. Urine flushes the urinary tract, mucus traps microbes in the respiratory tract, the mucociliary escalator then expels that mucus, tears wash our eyes out, saliva rinses our mouth out, and peristalsis moves material through the digestive tract. Without a way to attach, most microbes are removed from infection before infection can begin. Because of that, adhesion will allow pathogens to resist these defenses, allows them to remain in place, and allows them to establish and colonize that tissue. For many pathogens, attachment is one of the most important virulence factors that they possess. And there we go. So let's begin with UPEC, uropathogenic E. coli. After reaching the urinary tract, which we talked about is in the fecal to urinary tract mechanism, these bacteria face a major obstacle, and that's urine flow. UPEC solves this problem using structures called type 1 fimbriae. These hair-like appendages bind to receptors on the bladder's epithelial cells and anchor the bacteria in place. By resisting removal through urination, UPEC can establish colonization and begin the process of infection. Without these adhesions, the bacteria would simply be flushed away. Now, notice of importance is that these adhesins require specific proteins, and they happen to be present on the epithelial cells of the urinary tract. If they are not present, they also cannot colonize. Now, Neisseria gonorrhea uses a similar but distinct strategy. This organism possesses pili, which allow it to attach to mucosal surfaces in the urethra, cervix, rectum, pharynx, and conjunctiva. These pili help the bacterium remain attached despite mucus production and other host defenses. Because attachment depends on specific interactions between bacterial structures and host receptors, the tissue that gonorrhea can infect are again determined in part by where those receptors are found. So if that, just like with UPEC, if the receptors are not there, attachment cannot occur. Streptococcus pyogenes demonstrates that not all pathogens rely on pili for attachment. Instead, it uses surface proteins known as M protein and F protein. These proteins bind host molecules such as fibronectin and fibrinogen, allowing the bacterium to adhere to epithelial tissues. Although the mechanism differs from UPEC and gonorrhea, the goal is exactly the same. Establish a stable foothold on the host tissue before the organism is removed. Although these pathogens use different adhesion mechanisms, they can be grouped into two major categories, fimbrial and afimbrial attachment. UPEC and Neisseria gonorrhea primarily use fimbrial adhesions such as fimbriae or pili. These hair-like structures extend from the bacterial surface and bind specific receptors on the host cells. In contrast, Strep Pyogenes relies primarily on afimbrial adhesins, including the M protein and F protein, which are surface proteins that bind directly to host molecules such as fibronectin. Despite these structural differences, the goal is the same, and that is to remain attached to host tissues and resist removal by host defenses. Different pathogens have evolved different solutions to the same challenge of establishing colonization. One of the reasons we spend so much time discussing adhesion is that attachment itself is a virulence factor. A virulence factor is any trait that increases the pathogen's ability to cause disease. Without adhesion, many pathogens would never establish infection because the body is constantly removing microbes through mechanisms such as urine flow, mucus movement, tears, saliva, and peristalsis. By attaching to host tissues, pathogens can resist these defenses and establish colonization. Once colonization is established, the pathogen has the opportunity to invade tissues, evade the immune system, and eventually cause disease. In many cases, if adhesion is prevented, the infection never gets started. Pardon me. Today, we examine the second stage of pathogenesis, which is adhesion. We learned that attachment is essential because host defenses constantly work to remove microbes. We also learned about the different mechanisms in which several organisms can attach, including both fimbrial and afimbrial mechanisms. And although that they differ, they all accomplish the same goal, allowing the pathogen to remain attached long enough for infection to become established. In our next lecture, we'll examine what happens after attachment and explore how pathogens invade host tissues. With that, thank you for attending, and we'll see you at the next lecture.